Assalamu alaikum. Greetings to you all. And today in this video series actually, I'm going to read a scientific paper on cancer. The title of this paper is Press Pulse, a novel therapeutic strategy for metabolic management of cancer. This paper was published by the authors of this paper were Dr. Thomas Seyfried, George Yu, Joseph C. Maroon, and Dominic P. D'Agostino. Today, this video is the fourth part of this series. In this series, I'm actually reading the whole paper. So if you want to read or if you want to listen to the first part or the previous parts of this paper, please go back to the previous videos. So this is the fourth part, and I'm going to start reading now. Tumor cell energy metabolites from cannibalism and phagocytosis. Emerging evidence indicates that macrophages or their fusion hybridization with neoplastic stem cells are the origin of metastatic cancers. Reference 589, 121 through 124. Radiation therapy can enhance fusion hybridization that could increase risk of invasive and metastatic tumor cells. Reference 91, 125. Cannibalism and phagocytosis of cellular debris are well-known characteristics of macrophages and of myeloid cancer cells with macrophage properties. Reference 121, 126 through 131. Shelton showed that glioblastoma cells with myeloid properties could survive in matrigel, extracellular matrix material, in the absence of added glucose or glutamine. Reference 132. The gradual accumulation of lactate in the media suggested that glioblastoma cells survived through lysosomal digestion and aerobic fermentation of glucoconjugates present in the matrigel. Glioblastoma cell death occurred immediately following the addition of chloroquine, which neutralizes lysosomal acidity and digestion. Reference 132. Shelton's findings are consistent with the more recent finding of Camfrost et al. In showing that pancreatic ductal adenocarcinoma cells could obtain glutamine under nutrient-poor conditions through lysosomal digestion of extracellular proteins. Reference 133. It will therefore become necessary to also target lysosomal digestion under reduced glucose and glutamine conditions to effectively manage those invasive and metastatic cancers that express cannibalism and phagocytosis. Genome integrity and energy metabolism. Excuse me. Genome integrity and energy metabolism. Emerging evidence indicates that the function of DNA repair enzymes and the integrity of the nuclear genome are dependent to a large extent on the energy derived from normal respiration. Reference 134 through 142. Previous studies in yeast and mammalian cells show that disruption in aerobic respiration can cause mutations, loss of heterogeneity, chromosome instability, epigenetic modifications, etc. in nuclear genome. Reference 28, 141, 143, 144. A protracted reliance on fermentation causes oxidative stress leading to the production of reactive oxygen species or ROS, mostly through the mitochondrial enzyme Q-couple. Reference 145. In addition to their role in oncogenic signaling, excess ROS production damages mitochondrial function and can be both carcinogenic and mutagenic. Reference 146 and 147. The somatic mutations and genomic instability seen in tumor cells thus arise from a protracted reliance on fermentation energy metabolism and a disruption of redox balance through excess oxidative stress. We recently discussed how a transition from respiration to fermentation could account for Schendt-Gorgie's oncogenic paradox, that is, the process by which various provocative agents, radiation, inflammation, hypoxia, carcinogenic chemicals, age, germline mutations, etc., could produce cancer through a common pathological mechanism. Reference 2, 148. Mukherjee and Kearns also struggled to explain the oncogenic paradox. Reference 149, 150. All of these provocative cancer-causing agents damage respiration, thus forcing the cell to rely more heavily on energy generated through fermentation for survival. According to mitochondrial metabolic theory of cancer, the large genomic heterogeneity seen in tumor cells arise as a consequence rather than as a cause of mitochondrial dysfunction. Reference 2, 1428. A therapeutic strategy targeting the metabolic abnormality common to most tumor cells should therefore be more effective in managing cancer than would a strategy targeting genetic mutations that vary widely between tumors of the same histological grade and even within the same tumor. Human evolution and adaptive versatility. Rick Potts, a paleoanthropologist at the Smithsonian Institution, suggested that evolutionary success of our species has been due largely to the germline inheritance of traits that bestowed adaptive versatility. Reference 151 through 153. Adaptability was defined in terms of, number one, the ability of an organism to persist through major environmental shifts, number two, to spread to new habitats, and number three, to respond in novel ways to its surroundings. Reference 153. These characteristics were honed over millions of years and enabled humans to adapt rapidly to abrupt changes in the physical environment, including changes in moisture, temperature, food resources, etc. Adaptability to abrupt environmental change is a property of the genome, which was selected for in order to ensure survival under environmental extremes. Reference 65, 154. Potts' hypothesis is an extension of Darwin's original theory, Chapter 4, Natural Selection, and can be applied to the individual cells of the organism, which exist as an integrated society of cells. Reference 65, 154. The success in dealing with environmental stress and disease is therefore dependent on the integrated action of all cells in the organism. Further, the integrated action depends on the flexibility of each cell's genome, which responds to both internal and external signals according to the needs of the organism. More specifically, only those cells possessing flexibility in nutrient utilization will be able to survive under nutrient stress. Environmental forcing has therefore selected those genomes most capable of adapting to change in order to maintain metabolic homeostasis. Reference 65, 152, 153, 155. This concept was first discussed in relationship to the management of brain cancer. Reference 65. The widely held notion that tumor cells have a growth advantage and are more fit than normal cells are in contrast to Darwin's theory of evolution and also to Potts' theory of adaptive versatility. Reference 65, 153, 154. It is difficult to conceive how a random accumulation of somatic mutations could enhance the adaptability and fitness of cancer cells. It is important to recognize that mutations in p53, KRAS, and RAF impact negatively on mitochondrial energy efficiency, thus making cells with these mutations less metabolically flexible than normal cells. Reference 28, 44, 53, 135, 156 through 159. Indeed, activating mutations in KRAS target mitochondria, thus enhancing glycolysis. Reference 53 and 160. Enhanced glycolysis will make tumor cells appear more metabolically fit than normal cells in hypoxic environments. Reference 161, 162. Most normal cells, however, cannot grow in hypoxia and will often die in hypoxic environments due to respiratory failure. Tumor cells are more fit than normal cells to survive in the hypoxic niche of the tumor microenvironment. Hypoxic adaptation of tumor cells allows for them to avoid apoptosis due to their metabolic reprogramming following a gradual loss of respiratory function. Reference 31, 32, 162, 163. The high rates of tumor cell glycolysis and glutaminolysis will also make them resistant to apoptosis. Apoptosis, ROS or ROS, and chemotherapy drugs. Reference 163. Despite having high levels of ROS, glutamate derived from glutamine contributes to glutathione production that can protect tumor cells from ROS. Reference 164. As long as the tumor cells have access to the metabolic fuels needed for glycolysis and TCA cycle substrate level phosphorylation, or glucose and glutamine respectively, they will give the appearance of having growth advantage over most normal cells. Reference 2. According to Darwin and Potts, mutations that bestow a selective advantage are those that will enhance survival under environmental stress. If the multiple pathogenic point mutations, chromosomal rearrangements, and mitochondrial abnormalities confer a fitness or survival advantage to tumor cells, then survival under environmental stress and nutrient deprivation should be better in tumor cells than in normal cells. Reference 165. This is not what actually happens, however, when the hypothesis is tested. For example, when mice or people with tumors are placed under energy stress using dietary energy reduction or glucose restriction, many tumor cells die while normal cells survive. Indeed, the health and vitality of the normal cells improves with time under dietary energy reduction, while hyperglycolytic tumor cells undergo energetic crisis, triggering apoptotic death. Reference 166, 167. Support for this contention comes from studies of treating brain tumors with dietary energy stress. Reference 114, 168 through 174. It is clear that adaptability to environmental stress is greater in normal cells than in tumor cells, as normal cells can transition from the metabolism of glucose to the metabolism of ketone bodies when glucose becomes limiting. Mitochondrial oxidative phosphorylation is less robust in tumor cells than in normal cells, while glucose utilization through lactic acid fermentation is greater in tumor cells than in normal cells. Targeting glucose availability will therefore cause greater death in the tumor cells than in the normal cells. Mitochondrial respiratory chain defects will prevent tumor cells from using ketone bodies for energy. Reference 145. Consequently, glycolysis-dependent tumor cells are less adaptable to metabolic stress than are the normal cells. This vulnerability can be exploited for targeting tumor cell energy metabolism. Reference 160-163. It is also possible that therapeutic energy stress could restore the microenvironment, thus reversing abnormal energy metabolism and growth behavior in tumor cells not containing genetic mutations. Reference 19-175. In contrast to dietary energy reduction, radiation and toxic drugs can damage the microenvironment and transform normal cells into tumor cells while also creating tumor cells that become highly resistant to drugs and radiation. Drug-resistant tumor cells arise in large part from the damage to the respiration in bystander precancerous cells. These cells are often those that eventually become heavily dependent on fermentation for energy. The greater adaptability of normal cells than tumor cells to energy stress is predicted based on the theories of Darwin and Potts. Reference 154. Metabolic flexibility allows the organism to respond in a coordinated way to environmental stress and limited substrate availability. Energy stress will force all normal cells to work together for a survival of the organism. Reference 154. Pathogenic mutations and genomic instability will reduce adaptability and metabolic flexibility under energy stress. The greater the genomic instability in tumor cells, the less will be their adaptability to stress. This concept is similar to that of novels except in viewing genomic instability as a liability rather than as an advantage to progression. Reference 154-176. Because energy generated through substrate level phosphorylation is greater in tumor cells than in normal cells, tumor cells are more dependent than normal cells on availability of fermentable fuels, glucose and glutamine. Reference 94. With few exceptions, most normal cells shift energy metabolism from glucose to ketone bodies and fats when placed under energy stress from glucose deprivation, insulin deficiency, and prolonged fasting. This shift is the result of adaptive versatility and genomic stability, which is lacking in the tumor cells but is prominent in cells and tissues with robust mitochondrial function. Tumor cells will have difficulty surviving and growing regardless of their complement of genomic changes if fermentable fuels become restricted in the microenvironment. Ketone bodies and fats are non-fermentable fuels. Reference 177. Tumor cells have difficulty using ketone bodies and fats for fuel when glucose is reduced. Reference 57, 178, 180. Although some tumor cells might appear to oxidize ketone bodies by the presence of ketolytic enzymes, reference 181, it is not clear if ketone bodies and fats can provide sufficient energy for cell viability in the absence of glucose and glutamine. The studies in immunocompetent syngeneic mice and xenografts with brain tumors are proof of concepts that tumor cells are less adaptable than normal cells when placed under energy stress. Reference 114, 170, 171, 182 through 184. Apoptosis under energy stress is greater in tumor cells than in normal cells. Reference 170. The multiple genetic defects in tumor cells will reduce genomic flexibility, thus increasing the likelihood of cell death under environmental stress that would lower glucose and elevate ketone bodies. Regardless of when or how genomic defects become involved in the initiation or progression of tumors, these defects can be exploited for tumor management or resolution. Reference 12. That's where I'm going to stop on this part. This was part 4 of this series. The next will be part 5, where I'll start reading through the results part. So hope to see you in that video. Take care. Bye-bye.